Our first speaker is Dr. Larry Goldstein, who is widely considered to be literally one of the world's leading authorities in stem cell applications and advanced medical therapies. He's the author of a book, and I confirmed it as true. The title of his book is Stem Cell for Dummies. And so he has a trunk full of those in his car. He'd be happy to autograph them for you right after we finish today. He does, pardon, just for me, yeah, I got it, yeah. He does have a lab at the Sanford Regenerative Medicine Center, and he does research on Alzheimer's disease and some other related diseases. He has a PhD from the University of Washington, and he's also completed postdoctoral research at the University of Colorado Boulder and the Massachusetts Institute of Technology. Dr. Catriona Jamieson is part of our faculty. She's an associate professor in the Division of Hematology Oncology, and a matter of fact, just named to be the division chief of our newest division, Regenerative Medicine. And her research focuses on finding more selective, less toxic treatments for those diseases that she specializes in, leukemia and other bone marrow disorders. She completed her PhD in microbiology, her MD in clinical residency at the University of British Columbia, and she also completed a fellowship, a postdoctoral fellowship in bone marrow transplantation at Sanford University. So we're looking forward to your comments, and thank you for being here today. So this series of events that we've hosted, and we hope to have many more, called The Future of Care. And we're so pleased that you're able to join us today. And we'll invite you to every single one of our future ones going forward, and hope you're able to join us. You see the topics that we've covered here. And so just a couple words about our health system. One of the secrets of UC San Diego is this great health system, and so we did a strategic plan a little over a year ago and created a new mission and vision to help guide us. And lots of ways to describe what we're trying to accomplish in our health system. A couple of simple ways. Our goal is that no patient should ever have to leave San Diego to receive world-class health care. Our second part of our vision, unwritten, but we'll get to the written part in just a minute. The second part is that our clinical enterprise is well known locally. Our goal, however, is to match our national prominence and excellence in academics, where we are the 14th ranked medical school in the United States, and in research, where we are the 8th ranked medical school in the United States. For a medical school that just graduated its 43rd class, a remarkable achievement. And so our goal is to have that same national stature in clinical care as we do in research and academics, and you'll see some of the investments that we're making toward that end. So, U.S. News and World Report. Dr. Brenner refers to it as the Bible, and so we take it seriously. And so you'd see the history here. In 2012, we had 4 clinical specialties ranked in the top 50 in the United States. The U.S. News and World Report ranks 16 clinical specialties for all 5,500 hospitals in the U.S. So in 2013, all the great investments, all the recruitment of great faculty. We have 13 programs. In 2013, we have 10 programs that were ranked in the top 50 in the United States. This year we set another record. 11 of our 16 programs are rated in the top 50 in the United States by U.S. News and World Report. So we're thrilled. We were ranked number 5 in the state of California out of about 600 hospitals. And not that we're competitive, but if we had one more, we would be number 3 in the state of California. So we're working hard. The other 4 that we have, all of them in the top 100. So we've got some strategies in place to see if we can get 1 or 2 more in the top 50. I'm sure everybody else is trying to do the same thing as well, but we're going to try to beat them to it. So this commitment to excellence gets reflected in a lot of ways. Top 100 by Truven. Magnet status, less than 2% of the hospitals in the world have magnet status. Third, A rating from Leapfrog. Their 2014 grade just came out. You'll read about it next week. We're reason to be optimistic. More than 100 physicians in the top doctors in San Diego this year, and health grades, top 5%. We were just a shade away from being in the top 1% of all the hospitals in the U.S. And so that's kind of the backdrop for the great pride that we have as a health system, and that we are making very significant investments not far from here in the Jacobs Medical Center, recruiting new faculty, a new outpatient pavilion. But today's about research, and today's about a research set of accomplishments and accelerating that research into clinical care. And so stem cell is the topic today, and you see here a picture of Denny Sanford. And behind him is the picture of the Jacobs Medical Center and the Sanford Consortium, which he, of course, helped found it. And so he provided a magnificent gift of $100 million to UC San Diego to help accelerate stem cell research into trials, into treatment, and into cures for diseases that previously had been untreatable. A remarkable gift from a remarkable man. And you're going to hear more about what this means. We have a brief video to share with you that kind of helps put this in context.
The discovery of penicillin and the structure of DNA, the arrival of the polio vaccine, and the first sequencing of the human genome. The most important leap in medicine is the leap from laboratory to patients. And now, UC San Diego is poised to accelerate the next big revolution. Redefining medicine by taking stem cell research out of laboratories and into clinical trials. Uniting world-class research organizations across the Torrey Pines Mesa and beyond. Opening our doors to the most renowned scientists in the field and giving our patients new hope. A new chance, a new era. The Sanford Stem Cell Clinical Center at UC San Diego. The future of medicine is here.
So we had a lot of media coverage, as you would expect, about this magnificent gift from this very generous philanthropist. And we were so pleased that this got picked up nationally. And this gift, again, is designed to accelerate the development of drugs and therapies inspired and derived from current human stem cell research right here in San Diego to cure diseases globally. And so this is a commitment that not only Mr. Sanford made, but you'll hear more later in our presentation. We've also made a commitment. We've committed up to $200 million more on top of the $100 million gift from Mr. Sanford to provide this excitement, provide this momentum to translate this research into real clinical care. Here's a picture of Dr. Jamieson, and again, this is great exposure from the Union Tribune, and she'll share with you later the gift that we received, the grant that we received this week from the California Institute of Regenerative Medicine. They went through a process, a grant-giving process, to design and name 3 alpha grants, alpha clinics in the state of California. Initially, we were going to appoint 5. They only appointed 3. We were one of 3 health systems in the state of California, each to get an $8 million grant for the same purpose. And so to be recognized by the California Institute, really a remarkable achievement. And Larry and Kat will talk more about that. So here's this leveraging this power of the Stem Cell Clinical Center. We see here in the middle the stem cells program, and we have all of these programs around it. The Sanford Burnham, the Scripps Genome Center, the Salk Institute, all partners, all collaborators, all of them spoke on our behalf for the CIRM grant, all spoke on our behalf for the Sanford gift. A remarkable amount of cooperation in this town that wouldn't happen in a lot of other places. You see this Genomics Institute, La Jolla Institute for Allergy and Immunology, et cetera. And then at the bottom we have our health system and our health sciences division, all the scientists, all the power of the University of California behind this. And so we have 4 locations for this related work: the Sanford Consortium, the outpatient pavilion, which will be constructed next to the Jacobs Medical Center, the Jacobs Medical Center itself, and our laboratory, our advanced laboratory, which is actually in this building where we sit today on the ground floor toward the parking lot. So we made a very, very significant investment in this infrastructure, and we were just thrilled to be where we are today. So our next speaker is Dr. Goldstein, and it's my pleasure to welcome him to the stage. Larry.
Good. I should have Paul carry me around all the time. So let me get all the little pointers and all these other things working up here tonight and get going here. So thank you all for coming today and joining us. I just want to preface what I have to say by echoing something that Paul said. This scientific and medical community is one of the world's best, if not the world's best. We're actually a well-kept secret, if you will, in our own backyard. And I'll give you just a little bit of evidence of that. I was a tenured faculty member at Harvard, one of the exalted, the center of the universe by its own description. And I left Harvard to come here 20 years ago. I've had lots of offers to go other places, Stanford, UCSF, what have you, and I'm not leaving. This place is simply the best place to work that I can imagine. And in part, that's because of, thank you very much. Yes, that's good. And in part, it's because of the extraordinary collaboration and cooperation. I can talk to people about my ideas without worrying that they're going to run off with them. I can actually get people to help me with experiments. And I'll just tell you right now that problems in medicine are hard. They're not easy problems. They can't be solved by one individual working on their own, or one lab, or even one institution. And that's why the extraordinary cooperation in this community is so important for us to get to where we want to go. So, now then, ordinarily, scientists such as myself are boring. And we're boring for two reasons. One of the reasons we're boring is we forget that most people in our community speak English. And so we hurl jargon at them and words they can't understand. And it's really kind of this code that we use so that y'all think we're really smart. But it's not it. We just speak a different language. So that's one thing. The second thing is that at the end of the day, scientists like me are boring because we're too conservative. We don't tell you what we hope. We don't tell you what we want. We don't tell you what we really think we can achieve. We learn to be very conservative in our upbringing as young scientists. Well, maybe if this were true, we might be able to implicate the suggestion that something or other. And you're so tired of all of these qualifiers that you don't even get to hear the end of the sentence. So today, I'm not going to do that. I'm going to do something that I rarely get to do in public. And it's safe, and it's not obscene. And that is, I'm going to tell you what I think we may be able to achieve. I'm not going to guarantee that we can achieve it. I'm going to tell you what I hope. I'm telling you what I hope we can do here at this institution. And I hope we can do it with stem cells. Now, why are stem cells so important? And the answer comes from a film that was made in the 60s that had a young Dustin Hoffman as a graduate from college. There's a clue, right? So you guys even know what the question is. No, I didn't get to get there, right? So as I was going to say before you stepped on my lines, there were two important lessons from that film. One, you're probably not going to think of, older women can be very interesting. And I happen to know that this is true because I am married to an older woman, being an older man. But second, yes, indeed, plastics. So why were plastics so important in the 60s, and why was that such good career advice? And the answer is, in the 60s, virtually everything we made or used was composed of wood and metal, and not much else. Natural materials. Plastics changed everything. Everything in this room has a hunk of plastic in it. Now, if you fast forward to today, we are in the beginning of the era of biological plastics. Biological materials that can be used, in essence, to build almost anything biological in our bodies. And what these are stem cells. These cells, remember, cells are the basic building block of our bodies. These stem cells have two abilities that are important. Broad brush here. One, they can grow and make more of themselves if you feed them the right stuff. And two, if you send them the proper chemical signals, and I won't bore you with the names of those signals, if you send them the proper signals, you can induce these cells to make any cell type you find in an adult human. Brain cells, heart cells, kidney cells, you name it, we can either make it now or we're getting close to being able to make it. So these are biological plastics. We can, in principle, use them to begin making cells that we can use to replace cells that are lost or damaged in disease. And that's why they're so very important. and in particular, so important for the future of medicine. So up until now, stem cells were primarily used in the treatment of different types of blood disease, like leukemias. My friend Kat Jamieson may mention those. Stem cell transplants, bone marrow transplants, can be curative for leukemia, and that sets the bar for what we're trying to achieve. It tells us where we have to go with all the other kinds of diseases we want to treat with stem cells. And I'm going to give you two very specific examples of diseases where we hope, and we are working as hard as we can to get there, that we may be able to treat here at the Sanford Stem Cell Clinical Center as a result of the generosity of Mr. Sanford and the generosity of my two colleagues, David Brenner and Paul Viviano, and certainly the generosity of my coworker who puts up with a lot, Catriona Jamieson. And those two diseases are ALS, or Lou Gehrig's disease, and type 1 diabetes. So let me tell you a little bit more. If you have a diagnosis of ALS today, and I know my friend Christine Morley knows this very well. Her brother was recently afflicted with this disease. And those of us who have dealt with it can tell you it's awful. It's a disease where the neurons, the brain cells that control voluntary muscles, the ability that I have to move my fingers, to move my feet, to move around, those brain cells die. We don't know why, they die, and eventually we lose the ability to breathe. We lose the ability to speak. It is horrible. These people are trapped inside their bodies. Now, as of today, we have one approved drug that can be used to treat ALS. And that drug, let's be blunt, it's terrible. It barely works. It improves the lifespan of these patients 3 to 6 months. It's terrible. There are a lot of new therapies being developed in clinical trials. I hope that every one of them works. But I have to be realistic. Some are going to work better than others, and the ones we're working on I'm very hopeful for, because time really matters to these people. Because typically, the time from diagnosis to death is 3 to 5 years. Some cases, as fast, regrettably, as 6 to 12 months. So this really is nasty. So why stem cells? Well, what we think we can do is make cells in the spinal cord which will keep the motor neurons healthy for longer periods of time. Special cells called astrocytes, and we can make these from stem cells. And so we are in the midst of work to try to develop this therapy. Now, if we are successful, let's fast forward from today, 2014, to 2024, and what it might look like if we achieve what I hope we'll achieve at the Sanford Clinical Center. Well, so first of all, here's a shot from the sky of UCSD Health Science Campus on the East Campus. And you would start out with a consultation at the Stem Cell Clinical Center outpatient unit that is currently being constructed. We would take samples of your tissue, and those tissues would come over here to this building, CALM, where we will have diagnostic genomic sequencers and the ability to produce cells that are made under safe conditions to be able to put them back into a human patient's body. Having done that, we have a wing of the new hospital where our patients will be recovering after surgery in an operating room, also being constructed with the help of Mr. Sanford, where we will have MRI-guided surgeries to put these cells into the spinal cord of these patients. And of course, the hope is that these patients will leave and they will go about their business. Certainly longer than 3 to 6 months or 3 to 5 years. Now, of course, we take every yard we can get when we're fighting disease. But this is what we aspire to. And I think we can get there. It's going to take some luck, and it's going to take one heck of a lot of hard work. But I think we're up to the job. Second example, type 1 diabetes. So you probably know that type 1 diabetes is another disease caused by cell death. But in this case, it's the death of cells in the pancreas that produce insulin. And if you don't have those cells, you need to take insulin injections or have an insulin pump. And of course, if it's a young child, can you imagine having to give injections to your 2-year-old or 4-year-old kid? And of course, you meet adults who've had this for years. It's terrible. The disease management is complicated, and they have a normal life expectancy, but they have got to have a very carefully managed life. So again, why stem cells? Well, if it's beta cells that are missing in the disease, maybe we can make new beta cells and replace them. And so again, if we achieve what we hope to achieve, in 2024, when you show up here, we'll do your consultation and evaluation at the outpatient unit, same one as ALS. Samples come over here for help in diagnosis and evaluation, and then back to the hospital or the outpatient wards, because in an outpatient procedure, we will have, we hope, devices that will in essence be an artificial pancreas constructed from stem cells. And you'll hear more about that momentarily with a local company. So, how will we get there? I'll give you the outline, and then I'll turn it over to my friend Kat Jamieson, who'll give you a little more of the details. You can think of the Sanford Stem Cell Clinical Center as being an enormous funnel. And the idea, and what we are building, is a funnel that will take good ideas for clinical trials and therapy development from anywhere we can get them. I don't care where the ideas come from as long as they're good. And we will put them into clinical trials at the Sanford Center and try to develop them into FDA-approved therapies over time. And that's what ultimately is going to define success for us. That's what we need to do, and that's what I think we can do. So, let me turn the stage over to my friend, Kat Jamieson, who will tell you how are we going to get there.
Thank you very much, Larry. It's always difficult to follow Larry in any talk, or Paul for that matter, because you can see that there's one thing that really makes this place stand out, and that's not only a high IQ with the great people we get to work with, Larry and David and Paul, but also a very high EQ. Does anybody here speak Spanish? Anybody? Yeah? Do you know that word cariñoso? That's here. It's an emotional intelligence that's a little bit unique. And that strong sense of community is something that you don't see in a lot of places. That sense that we should all be able to know about this stuff. You know, that this isn't private information, this is our information, and that we're going to get there together. So we're not really talking so much about things in the future, we're talking about things now. And the reason we can talk about bringing some of these things into the clinic now is we have the boots on the ground kind of strategy, where we keep our feet on the ground. It's not really about self-aggrandizing stuff where you say, hey, I've got this great reputation. We say, let's work together. I'll do what I can, you do what you can, so the physicians and scientists actually work together. But the third part is our patients work with us. And I just came from clinic, and boy, they tell you when you get it wrong. You know, they say, yeah, you fixed my leukemia, but what about this basically a neurodegenerative disorder that I can't quite deal with? What caused this? How do I make this better? You fixed my leukemia, but what about my blindness? I can't see you. You know, are you going to fix that? So we've made great strides in some ways in dealing with things because we can sample the blood or sample the bone marrow, but we need you to help us fix bigger problems that affect more people. I'm not saying forget about leukemia, because I think that's really important, but I think stem cells can help us to understand and potentially to fix some pretty bad disorders that before we thought were untreatable. So before we were talking about you know, a movie star that had stem cell treatment. So a lot of people think about stem cells and they think about rejuvenation. Yay, I'll be younger. But I say youth starts from the inside. Part of that is attitude, but a lot of it is we need to be healthy on the inside. We need to make sure that we can not just repair tissues, but regenerate stem cells and give them the cue that they need to do their job. Sometimes they can't do their job. You know, I was a bone marrow transplanter. You can even replace the bone marrow with somebody else's bone marrow, but it doesn't always work, and a lot of people don't have that as a choice. The reason I left bone marrow transplant is 20 to 40% of my patients would die in the first year. And I didn't know who it was going to be. So that was kind of tough because you feel a very strong sense of responsibility for that person. So we thought, okay, we need to get back to the bench. Then I came here in 2005 to give a talk at the Salk. And other than the phenomenal view, I looked, when I got back into the auditorium at the people. And I saw Larry was there, and there were all these people that were actually willing to work together. And that doesn't stop in the lab. It doesn't stop in the clinic where people are willing to roll their sleeves up and say, how can I help you? The patients say, I want to be part of this. I want to be educated about this. I want to educate you about this. And that's what's uniquely different. So when I do clinical trials, they say, why are your clinical trials going so well? I say, it's not me. The patients who are involved are like our astronauts. So I gave a talk at this Inspiring Minds meeting, not feeling particularly inspired until I looked around and I thought, wow, people here are pretty optimistic. They don't have that sense of nihilism or cynicism that you get in some other places. And so the person in the audience that kept asking me a lot of questions had actually been involved in the Apollo program as a rocket scientist. But he said that program wouldn't have gone anywhere without the astronauts. Somebody had to be willing to take the risk, and that's what patients do on clinical trials. They take that risk. They're willing to help other people, not just themselves. So that's what's unique. And this is going to be incredibly important as we bring stem cell therapies to the clinic. So we know we can regenerate certain damaged tissues from 40 years of bone marrow transplantation, but can we do that for other tissues? Can we help people with diabetes, as Larry was alluding to? Can we help people with terrible diseases like ALS? And so basically we have different sources of stem cells. We have adult stem cells. Bone marrow is a common source. We can take our skin cells, thanks to Shinya Yamanaka and others, and make what are called induced pluripotent stem cells out of those, and then make them grow up into different kinds of cells. We have very primitive stem cells. We can do the same thing. So we think we can take these cells and regenerate damaged tissues. But the Food and Drug Administration said, hey, be careful. We want to be safe first and foremost. And so before we can take any of these stem cell-derived therapies to the clinic, we have to get what's called an investigational new drug application. Has anybody filed an IND before? Very huge. We just did one a few months ago. It's a colossal amount of work, but it's actually a worthwhile experience because it makes you think about safety. The one thing we have to be aware of, though, is primum non nocere. So when you take the Hippocratic Oath, that means above all do no harm. So we have to have this balance where we remember that the diseases cause harm. And that we have some pretty draconian diseases we're going after, so we have to come up with therapies, but hopefully therapies that are fairly safe. And this term precision regenerative medicine means we have to give the right therapy to the right person at the right time. So it means having better diagnosis, better ability to predict how the disease is going to do, and something that actually works instead of nickel and diming people with things that work for 2 weeks. We need something that's more durable. So we do this in the context of clinical trials. Has anybody here known anybody or been involved in a clinical trial? Yeah? Yeah, okay, so people know a little bit about this. So phase 1 is just safety. If we give a stem cell product, is it safe? Can people tolerate that? And then we get an early idea of, well, maybe it actually might work. And so we're seeing more of that recently, where the science is better going into clinical trials. Phase 2 is efficacy. So here we see, is it effective? Does it actually look like it's helping? And then phase 3 is it more effective than our current standard of care? And that's how we get through the clinical trials. And then usually the drug, device, biologic, small molecule is approved and then will become used for that indication. Now we have some pretty tough diseases that we want to go after because they affect people. Now I worked on cancer for a long time, so that's not so hard for the FDA to say, okay, we know cancer tends to have a shorter lifespan, especially leukemia. Especially leukemia in people who are, in their 70s, people don't live that long. But what about spinal cord injury? That's a terrible, devastating thing that happens to people, often in their 30s. This is a huge proportion of Americans who had a motor vehicle accident, they were rear-ended, they end up with a spinal cord injury, and there's really very little to do. So this is terrible quality of life-wise, it's also terrible quantity of life-wise because people have a lot of subsequent issues and extremely expensive for our healthcare system. And so if people are quadriplegic, meaning they can't move anything below their neck, you can imagine the kind of resources they're going to need to be able to get around. So we were very fortunate here to have fantastic colleagues, Martin Marcella, and then Larry Goldstein was involved in this, as well as Jennifer Braswell. and some other people who said, we think we can bring this to the clinic. Denny Sanford's gift helped a lot with the attempt to get neural stem cells into the spinal cord of people that have had spinal cord injuries. And so this neural stem trial is basically predicated on preclinical models that suggest that you can repair that defect in the spinal cord. So here, there is a defect in the spinal cord that happens after injury, and this is after the stem cells have gone in and they basically fill the defect. It's like putty. They're going in, they're filling the defect, and then the nerves go, okay, you're right next to me, maybe we can actually communicate again. So that gets rid of the spasticity. So a lot of what happens with spinal cord injury is the limbs get really stiff and people can't feel anything. So in the preclinical, so before the clinical trial model, there was an increased sense of touch. So you may have seen this in the news. The neural stem trial started last month. This is a phase 1 open label trial, meaning that we know what we're giving the patients. It's happening here. Joe Chiacci is a fantastic neurosurgeon and he's running the trial. The planned number of patients is 8, and these are for people that have had thoracic spinal cord injuries, very, very common. So they get injections into the spinal cord, which is completely defective because they had a huge injury there, of these neural stem cells. And so basically it's to assess safety. And this is the first operation that was done. That's Joe putting the cells into the spinal cord, and Martin is off on the side. He's got the cells prepared. So we're not talking about the future, we're talking about now. We just want to be able to do this kind of therapy for other diseases that are even tougher, as Larry alluded to. Larry likes a challenge. I can attest to that. ALS is a really tough challenge, so we want to see if we can move it on to thereafter. And then for diabetes, there's a local company called ViaCyte that had made really clever technology, and they said, we're going to go after diabetes because that's $240 billion spent annually for our healthcare budget on diabetes. This is where people have chronically elevated blood sugar, they can have eye problems, heart problems, you name it. People can have big blood vessel problems, nerve problems. Affects 347 million people worldwide, but 100 million actually require insulin, and this is 14% of the U.S. population that are diabetics. 1 to 2 million have type 1 diabetes, meaning they have to inject themselves with insulin, usually many times a day, and that gets kind of tiresome. So it doesn't even work some of the time. So basically, ViaCyte had this idea to make a renewable source of cells, so these are stem cell derived, and they know how to make insulin, provided you implant them under the skin in a special device called the encapture device. So that allows the cells to be in the body, under the skin, they get a blood vessel supply, and then they start making insulin. And so that clinical trial also started within the last couple of weeks, and the first patient was discharged after having implantation of these things underneath the skin in the back. We're the first site, so the UCSD patients, there will be 3 to 6. They basically have these devices that are about that size, so they're not very big, maybe a couple of times the size of a nickel, very thin, they're going to be under the skin, and then just a couple of small devices as sentinel devices. So the first in human trial started here. This is Bob Henry running that, and very, very well attuned to the disease. And again, our patients matter because they're also very well informed and they want to be part of the process. And then lastly, what about cancer? What about stem cells that go awry? We've talked about good stem cells that will go in and do their proper job and repair the tissue. What about bad stem cells, cancer stem cells, that are the root cause of relapse? So if we think, the #1 cause of death in this country for people under 85 is actually cancer. That means most of us have a family history of cancer, or one of us will get it. So we got to do something about it. So we started working on the idea that cancers frequently think that they're embryonic. They're very primitive, particularly when they're in their most aggressive form, or in the case of leukemia. So Tom Kipps discovered at the Moores Cancer Center that 94% of people with chronic lymphocytic leukemia, the most common blood cancer here in the US, express an embryonic protein called ROR1. He said, you're going to roar, we're going to roar back. So basically with a CIRM grant, the California Institute for Regenerative Medicine said, yes, we're going to give you money, but you have to be very focused, and we want you to make one product. And the best product we think you can make is an antibody to target that protein, because we think that's going to be important. So we called it CIRM-tuzumab, to reflect CIRM. And we started this clinical trial last month. I saw the second patient on the trial today. And he's going, what's all the fuss about? And so, you know, he thinks it's not that big a deal, but he's grateful to have it. This is the syringe for the first patient that got that antibody. And basically the idea is we'll see if we can get rid of these cancer stem cells that we think are the root cause of relapse. Tom Kipps is the IND holder and I'm the principal investigator of the trial, with Mike Choice, Ravina as the co-investigator. So I think the most important thing for today is that you're here. This is a privilege to be able to talk to you. You can make a difference. And Pam Werner, who in the back, has made a difference, yes. So we've got a couple of drugs to clinical trials because you said, I think we need a little fix here. I think we need an installation of hope. And this is an example of that, where actually this patient of mine, or she wasn't my patient, she was a local community oncologist patient who said, you know what, I heard you have clinical trials, and I have this person who I know has a life expectancy of about 2 months. He said, oh yeah, it's 6 o'clock, it's a Friday, I don't know. We do have a clinical trial, it's just opening. I don't know if we'll be able to get her on. He goes, you just have to meet her. I'll make the insurance work, just get her in your clinic. I said, okay, we'll get her in. And he said, you know, she has a goal in life, and she'll talk to you about it. So I met Linda. I said, what's your goal? She said, I want to meet my grandkids. I said, well, how old are they? Or she said, I want to be able to look after my grandkids. She said, they're not born yet. Oh, we've got some work to do here. So I just wanted to show you this story because this is about targeting these cancer stem cells with therapies, with one particular drug that was developed locally that was on the shelf at a local company called Pfizer. They weren't going to develop it, but because of philanthropic support, funding from CERM, you know, you piece all the things together. She had a chance. I just didn't know it would be as good a chance, but I wanted to have her say what she thinks.
This is a symptom.
This bruising is one reminder of a disease that turned Linda Lowe's life upside down.
I'm bald and bruised, but I'm still fighting.
More than 2 years ago, the bone marrow disease living in her body, MDS, the same one affecting Good Morning America's Robin Roberts,
I literally fell down and couldn't get up.
turned into leukemia. Doctors told her she had a short time to live, likely months. Her condition was too grave for a bone marrow transplant. Her search for answers led to the UCSD Moores Cancer Center and a clinical trial born from a unique annual symposium, mixing academia and the pharmaceutical industry. Because of the bonds formed, researchers called up Pfizer to compare notes on a new drug aimed at other cancers.
Together, we were able to reach a conclusion very quickly that this drug may be very effective in leukemia.
Just a year later, fast for the research world, Lowe was on an experimental drug that blocked cancer cells from cloning themselves. A year later, she qualified for a bone marrow transplant. She is now in remission.
I don't know that that would have happened if this collaboration had not taken place.
So the collaboration was with people like you that actually thought they could make a difference, and they made a difference for Linda. She's now cured. She's got 4 grandkids. They keep her pretty busy. You know, I don't think she would have had a chance to have a bone marrow transplant without that incredible foresight and generosity from people just like you. So I wanted to thank you and let somebody else take over, but thanks very much.
So when we say we're redefining the future of care, we mean that literally. This translation of groundbreaking research to real trials, to real treatments, to real cures happening today, only in an academic health system. So we have an opportunity now to learn a little bit more about this. We talked about how expensive this is. Our 10-year budget for this is about $300 million, and what a commitment this represents. So we have one more video, and then we're going to do some questions and answers. We're going to bring our panelists up, and you'll have an opportunity to discuss the things that are of interest to you.
Every time you have the chance to be the first to do something, to set an example by what you do in word and deed, we have within our power the possibility of changing the course of diseases that currently just kill people. The time has come to start working with human volunteers and to begin testing therapies that work in animals with stem cells to now bring it to therapies that we hope will work in people.
We're going after some pretty devastating illnesses that really haven't had any kind of hope for treatment before.
The hope for these diseases is that this research will lead to an actual cure and a treatment for countless patients, not just here in San Diego, but worldwide.
We can think of stem cells as being the biological versions of plastics. We can use these to make any cell type. And if we can make new ones, then we can learn to replace those cells that are lost or damaged in disease.
It will help us to devise new therapies to expand stem cell populations and come up with not only new diagnostic techniques, but new therapies. The infrastructure is here to really expand on stem cell research.
The Sanford Stem Cell Clinical Center, this summer, we hope to be launching our first stem cell clinical trial.
We're creating these new facilities to complement this research. We will have a dedicated unit in the Jacobs Medical Center for stem cell therapy. We will have a dedicated outpatient facility, steps away from the inpatient facility.
So this seamless transition between the lab and the clinic is the reason that this is the right place to do it.
The fantastic news is our hospitals are embedded in one of the top scientific regions in the world.
We're ringed by 600 biotech and pharma companies, and we're one of the biggest clusters of biotech in the U.S.
We live in this very rich intellectual atmosphere where people are free with their ideas. Institutional borders don't mean very much.
The basic scientists and the physicians talk to each other more here.
And La Jolla, San Diego, is one of the very few environments in the world that has these kinds of unique features.
No place else is it happening, right here in San Diego. It's remarkable, doesn't even come close to describing it.
Individuals do make a difference, and Denny has done an amazing thing.
This is not just a $100 million program that Denny Sanford was so incredibly generous to create. This is another $175 million on top of that UC San Diego is committed to this.
We have a unique approach to these problems. We have a unique scientific and medical community, and we have a leadership that can bring these factors together and program the future for success.
I can't wait to get started. This is an opportunity that we've been waiting for a long time.
This is a great investment from the perspective that patients that have no possible way to cure or treat their disease. This will provide not only hope, but a viable opportunity for a cure.
We need your help. We can't do this on our own. We need more than just one Denny Sanford. We need multiple Denny Sanfords. We need people who are going to help us by donating small amounts, large amounts, whatever it takes to win this battle. If we don't try, we're missing the greatest medical opportunity we've ever had.
We're going to invite Dr. Brenner, Dr. Goldstein, and Dr. Jamieson to the stage, and we'll be happy to address any questions that you might have. We do have only one ground rule. If you do have personal, specific questions about your clinical condition, we'd be happy to talk with you privately later. But we'd like to keep the questions kind of high level. So any questions that you have, please, Tom.
You mentioned that we've started a couple of clinical trials for stem cell therapy. Are there other clinical trials underway, or have there been any completed on stem cell therapies to use the services?
I'll start this and then Ken will chime in. So, yeah, good. So the question is, what's in the pipeline and how far have we gotten? So in the area of bone marrow transplant, there's a great deal of completed work where we know a lot about different ways of using those kinds of stem cells, although still there's a lot of room for improvement. In terms of clinical trials for all the other ailments we're talking about, we really are at the dawn. We are just now beginning with cells where I think we can really make a difference. Now, I just want to caution you, there is what we sometimes call snake oil out there, where people will tell you, oh, give me $50,000 of your money, I'm going to shoot you up with who knows what, here, or Tijuana, or Bulgaria, or wherever. Watch out for that stuff, because the legitimate stuff, the work where we can really tell you this is safe and this is really going to work, it really is just beginning now. Now, in addition to the 3 we've already launched this year, so diabetes, spinal cord injury, and cancer. Wait, it's a big job. We have a pipeline, and we have another half a dozen to a dozen that we think we can launch in the next year to 2 years. And when I said we want the best ideas we can get wherever they come from, I'm not kidding. We sent out a couple weeks ago an email, literally, to every investigator on the La Jolla Mesa who we think has some interest in stem cells, and there are hundreds of them. And we basically said, come to us with your proposals. Let us look at your ideas. We're going to pick a few, and we're going to help you get them done. So, Kat?
Yeah. The other thing I wanted to mention is when I was at Stanford, I would go for coffee desperately in the morning. And it was at the DNA Cafe. And I was always beaten to the front desk to pay by Paul Burke, who discovered recombinant DNA. And he said they couldn't have foreseen all of the potential opportunities from recombinant DNA. He actually thought it would be gene therapy, but actually we got all these factors, growth factors for the blood. We got recombinant insulin. We got all these things we never expected. That's what we need to think about with stem cell research. We're going to learn how our normal tissues regenerate themselves. We may be able to boost that capacity, whether it's for cartilage or some other tissue in the body. So it's not all just about cell replacement therapy. We want to learn how we regenerate tissues and do that in a safe way. So the reason it's so important to do this in an academic center is we will be very rigorous about how these trials are done. We're not going to be drinking our own Kool-Aid. We're going to be making sure that we're measuring responses, and we're very careful about how the work is done. We'll know if it worked, why it worked, and if it didn't, why it didn't. So we've got a lot of things in the pipeline. And we look forward to receiving your ideas as well.
Do you have a question, ma'am?
Yeah, I want to know, when you implant for diabetes 1, what is going in? What is it made of?
Yeah, so basically the question is, for the Encapture device, what is it made of? And this is basically getting back to what Larry was alluding to, that the stem cells are like the plastics of the future. But the actual device is just like a porous, almost plastic membrane. It has little holes in it. Think of the sandwich bag you put your sandwiches in, but it's got tiny little holes in it. And it's a very, very, kind of like that, very thin device, and it's got cells inside. So the cells can't get out, but what they make can. So what they make is insulin. And so they're very small devices, 2 times the size of a nickel. So 2 nickels, put them together. It's just a little thicker than that. They go under the skin in the back. They'll stay there for 2 years, and we'll see whether people are producing insulin in response to eating, is it appropriate response to blood sugar levels. And that's basically the point of the trial. Are these little devices safe?
Last month?
Yes, last week. Yes.
First patient.
Yeah.
I just want to add that this is a remarkable partnership, that a lot of the initial intellectual property was developed at UC San Diego, how you differentiate the embryonic stem cells into beta cells that produce insulin, that the company Biosite is a San Diego company, that the support for this trial was funded in part by grants from the California Institute for Regenerative Medicine to Biosite. And what really pushed it over was Larry and Kat, the people at the Sanford Stem Cell Center, to provide additional support through philanthropy to do this, so that the first patient studies will be done here. So it's really an incredible, great San Diego story that everyone contributed, the whole community contributed to make this happen.
Yeah, I wanted to mention a little bit about that synergy you're talking about, because David's been very self-deprecating as usual, as is Paul and Kristen. So this was only possible because of that gift. It did make a big difference in terms of being able to get the Biosite trial to the clinic, and their leadership, to be able to see into the future. A lot of people are just stuck in the present, and it's hard to get out of your comfort zone and say, we need to change this. We need to change how we treat type 1 diabetes. So that leadership is key.
Lots of questions. Yes, sir.
Were there animal trials ahead of this? I mean, it just seemed like we jumped right into the books. I'm just curious with Larry why he picked ALS, which doesn't seem to hit a very large number of folks in the society, versus something else that would make a bigger difference.
Okay. So 2 really good questions. So question 1, did we do this to animals before we did it to people? Absolutely.
Excuse me, can you speak up a little bit?
Yeah. Hold it, okay, is that better? All right. Sorry. Let's see, should I go back half an hour? So the question was, did we do this to animals before we did this to people? And the answer is absolutely yes. Many rats, mice, and pigs died before we even touched a human.
No, they were cured of diabetes.
Well, so you can see why this guy is the dean. He's faster than I am with a punch there. So and in the case of type 1 diabetes, for example, if you are a mouse with type 1 diabetes, we can cure you. Absolutely. If you're a rat with a spinal cord injury, we can make you a lot better. So we've done that. But now, as I like to say, humans are not just big mice, rats, or pigs. We're different. So that's why the human trials are so important, and why we kind of hold our breath when we do the very first patient. You just don't know what's going to happen. You know, we did all our homework. Second, why ALS? The answer is twofold. Well, threefold. One is, if you meet an ALS patient, you want to do something to help them. That just goes without saying. Second, when you're developing experimental therapies, particularly new ones where you don't know what all the risks are, it does help to work, in a sense, on people who have terminal diseases. They have a very high risk threshold, and when we do clinical trials, it's very important that we balance risk and benefit for any given patient or collection of patients. Once we know something about whether a treatment or a cell or a drug is safe and may be effective in a rare, very dangerous disease, then we can start to branch out to diseases that aren't as severe, where the track record in the severe case really helps us. Yeah.
We have time for 3 more questions. Yes, sir.
What's your feeling on aphasia for diabetes?
Sorry, I didn't quite catch on what?
On aphasia?
Let me know a little bit more about that.
It's something Sanofi's going to market for mankind.
Mm-hmm.
It's aphasia. It's an inhaler.
Oh, yes. Yeah, so there were a lot of, so the question is, what about inhaled products for diabetes, like inhaled insulin? That was a big strategy a few years ago, and it may be that they've improved the technology. Basically, what happens with our lungs is we kind of need them to breathe. So a lot of these inhalational products were really irritating. Even if they didn't basically damage the lungs, they made people feel bad. And so the other attempts to have inhalational products didn't work very well. Maybe they found something that's less irritating. So it would be great if people could absorb insulin and be able to take it up adequately. Our lung capacity is actually a good way to get to those blood vessels to have insulin well absorbed. It may not be as readily titratable. So with this device that's implantable under the skin, we think that will probably be more physiologic instead of getting this big boost of insulin all of a sudden, where people can really drop their blood sugar. So we know that when people drop their blood sugar too steeply, that can be bad. So we think it's going to be a more measured, reasonable, appropriate response to have these devices.
Paul? I think at the outset, with stem cells, the public was terribly misled. We came away thinking that within weeks or months there's going to be an absolute cure for everything that afflicts the human species. This, of course, was wrong. I'm wondering how you overcome that, because I think it still exists in some cases. The other thing is, I was wondering, and the question about ALS, when you're dealing with that, it seems to me it will have other benefits with other forms of nerve damage and nerve problems as a result of your research. So those two things.
Yeah. Good. So the first question is, how do we deal with what did develop in some quarters, I'm afraid stoked a bit by the media, about overhyping. And you know, it's a fair question and it's a fair issue. At the end of the day, the best I can do is tell people the truth. And you know, folks like you who are here to listen, that's great. We keep telling it to the media and the press, and we will continue doing that. We can't always control exactly what they write, and they look for drama, let's say, in many cases. So, you know, I think we just have to keep doing what we do. We tell the truth. The second question, or comment really, is, you know, will the fruits of our ALS work develop into benefits for other diseases? And you saw the heads nodding here. Absolutely. So there are many other diseases where motor neurons die. There are many other diseases where different cell types in the brain die. And the kinds of strategies we're developing for ALS are absolutely going to help us with the others. No question about it.
Yes, sir. Last question.
I'm a former practicing physician in this community, and stem cells, when I went to medical school, were something that you saw in bone marrow examination.
Still true.
But the most important thing that I saw today, and the most important thing that I think San Diegans should appreciate, is the one slide that you had, where you had stem cells in the middle and 9 or 10 little things around them. We are really, really fortunate. We are an integrated medical research community. As you said, Doctor, people are not afraid to share information here. It's a collaborative thing. And if we're going to have success, and we will have success, I'm 100% on that, it's because people will share. And in this community, that's why you left Harvard.
It's true.
That's exactly why you left Harvard.
And the weather.
No ivory towers out here. There are 55 ivory towers, but they all talk to each other. It's the most important thing that the UC campus has been able to do, and my friend Bill Otterson, who started Connect, made it happen. And it's so, we are just so fortunate. It's a very exciting thing to hear what's going on here, and I congratulate you on your work.
Thank you.
As you can tell, we are incredibly blessed to have world-class, world-renowned physician scientists. Please join me in thanking Dr. Goldstein. They will be here at the top side.